Looking, of course, at maybe what was sort of a headline outcome, at least stressed by the authors, of course, being the immune response and the emphasis being on a greater rate of patients versus in the Oxford vaccine talking about T cell and immune response, antibody response, sorry, being 14 and 28 days respectively, I think. I was curious if you could, I guess, share some, light on exactly what our barometer looks like for what we want there, and what exactly is a proper gauge. Are we looking for just a greater rate of course, antibody response, or are we looking for something that moves faster? I also know there was some expressed concern in the Oxford trial that maybe they were seeing early signs of the need for a booster shot, which of course, is an open discussion with all of these candidates right now. What exactly are we looking to accomplish in terms of the responses?
Yeah, so I think that's a really good question. And this is also where the efficacy trials become really important because it's very difficult to predict what level of immune protection you're going to get based on antibody titers alone. So typically, depending on the pathogen, depending on the type of vaccine, you'll get those IgG neutralizing antibody titers anytime from 7 to 14 to 28 days later. And sometimes, yes, it does require a booster shot. It appears that the Moderna vaccine will definitely require a booster shot. It's a little trickier with the Oxford vaccine because, again, they were able, with a single shot, to elicit titers of neutralizing antibodies as well as antigen-specific T cells in most of the patients. It really remains to be seen what those declining antibody titers later on might mean. You may need to have a booster shot, and that's something that normally is established in phase 2 or phase 3 trials in which patients are monitored for a much longer period of time than we're doing in any of these clinical trials. So one of the reasons why vaccine development normally takes so long is that people in these different trial groups are monitored for long periods of time, in part to see how long that immune response lasts and to see if you do need a booster. In this case, we're basically making assumptions on, antibody titers, and in the case of the Oxford vaccine, T cell immunity, but we don't really actually have any idea what that means in terms of long-term protective immunity. That's normally something that we would determine before the vaccine gets approved. In this case, I think we're going to be determining that as we go, because I suspect that as soon as any of these vaccines show some real concrete signs of efficacy in phase 3 trials, they're going to go ahead and approve the vaccine and start gearing up to distribute it widely. Unless, of course, there's evidence that these adverse effects may be more common than they appeared to be in the phase 1 or phase 1/2 trial. So I think it's really a big open question what this means. Certainly, at least if we're just looking at antibody titers, the Moderna vaccine appears to definitely require a prime boost regimen. Whether that would, for longer term immunity, whether that would require another boost, which actually might increase the risk of having adverse events, being that most people reported adverse events after the booster shot and not after the initial prime shot. That's something that we're going to have to kind of figure out as we go along. For the Oxford vaccine, many live attenuated vaccines or viral vectored vaccines are usually one dose vaccines because they do actually have a replicating virus in there that's thought to trigger longer term, longer lasting immune responses. But again, this is kind of a wait and see situation because it is so unprecedented. And we're approving these at a greatly accelerated rate compared to what we normally do.